Okay, hi, welcome back everyone. Thank you for joining us for today's presentation entitled Spatial Mapping of Gene Expression in Tumors using the RNAscope Technology. And it's my real pleasure to introduce you to Anushka Dikshit, application scientist from Advanced Cell Diagnostics. And for her complete biography and all our speakers, you can visit the tab at the bottom of your screen for more information. Now, if you have any questions during this presentation, we encourage you to submit them in the Q&A box, and our speaker will respond back to you via email. So please join me in welcoming Anushka. I'll turn the presentation over to her.
Thank you for being here today. Today I'll be talking to you about the RNAscope technology, which is a gene expression technology that allows you to visualize your targets with spatial and morphological resolution. For those of you who are not familiar with the RNAscope technology, it is an in situ hybridization platform that was specially designed to overcome some of the traditional limitations of the ISH assay while still maintaining high sensitivity and specificity of detection. The RNAscope assay is perfectly designed to perform gene expression analysis with spatial resolution in really complex and heterogeneous tissues like the tumor, where you have different cell types interacting with each other. Sorry, my slide just went on its own without me clicking anything.
That's okay, huh? I wonder what happened. Cindy, you didn't touch it, right?
I may have.
Cindy didn't hear. Okay.
I may have. I'm sorry.
That's okay.
I'm looking at your presentation in PowerPoint, and I think I might. Sorry. Sorry. I'm going to put it away now.
That's okay. Anushka, you can start from the top of slide 3. Was that the one that you were on?
I was in slide 3, yes. So can I start from there?
Okay. Yes, yeah, definitely. Let me make a note really quick. Give me just a second.
Sure. Yeah, sure. Yeah. My timer. I think I added 4 laps. How did I do that?
Oh no, okay. Okay. All right. So give it just a few seconds and then you can begin.
The RNAscope technology is perfectly positioned to visualize gene expression analysis with spatial resolution in really complex tissues like the tumor. A heterogeneous tissue like the tumor has different cell types that are interacting with each other within the tumor microenvironment. So to study the function of the different cell types present within the tumor microenvironment, it is essential to use a highly sensitive technology. The 3 major parts of the RNAscope technology are the target probe design, where ACD will design a specific target probe against the mRNA transcript of your interest. The signal amplification strategy, which includes a proprietary amplification system, which allows very high signal-to-noise ratio. And a detection and quantification system, which allows visualization of a single molecule of RNA at single cell resolution. So this assay can be applied to a wide variety of target types, which include mRNAs, long non-coding RNAs, splice variants, homologous sequences, short targets, point mutations, and so on. Additionally, our assay is flexible and can be made applicable to FFPE tissues, fresh and fixed frozen tissues, cultured cells, and PBMCs. Next, I want to talk to you about the 2 key features of our technology, which are the probe design and the signal amplification system. As you can see on the screen, our probe is depicted as a single Z molecule to mainly highlight the different regions of the probe. The target binding region of the probe, which is the bottom part of the Z, binds on to your target transcript of interest. The preamplifier binding site, which is the top part of the Z, is where your amplification tree will form, and the linker region connects these two parts of the probe. Now we design our probes such that two Z probes bind in tandem onto your target sequence next to each other. Once they are bound, you have your preamplifier bind on top of the Z pair, which is followed by binding of amplifier and labeled probes. Now these can either be chromogenically or fluorescently labeled probes. So this amplification strategy allows for very high sensitivity of detection and detects low expressing targets as well. Now, how do we ensure high specificity? As I mentioned earlier, the two Z molecules the two Z probes have to bind in tandem next to each other. If a Z probe does not bind specifically to the target sequence, then the preamplifier cannot bind onto the Z probe and there won't be any signal detected. As a result, this allows for high signal to noise ratio, ensuring high sensitivity and specificity of detection. In addition to our RNAscope assays, we also have the BaseScope assays, which are specifically designed to visualize short targets such as exon junctions, highly homologous sequences, and point mutations. For our RNAscope assays, we design probes as a pair of 20 ZZ probes, which is a pooled pair of probes. And this is again mainly for targets that are over 300 nucleotides in length, whereas our BaseScope assays are designed for short targets and therefore have 1 to 3 ZZ pair of probes. And this is mainly for targets that are between 50 to 300 nucleotides long. In this slide, I have described the breadth of our assay portfolio, which gives you a sense of how flexible we are when it comes to plexing and sample types. You can either perform single-plex analysis or duplex or up to 12-plex for target detection. So for 12-plex, you can visualize 12 targets simultaneously on your sample of interest. Now, the 12-plex assay is applicable for fresh and fixed frozen tissues. Our assays can also be performed both with manual or automated platforms. And currently, we have over 25,000 catalog probes that are available to use. In addition, we can also custom design any probe you want as long as we know the target sequence of interest. In the next part of my presentation, I will talk about the RNAscope workflow in combination with the GeoMx DSP. And during that part of my presentation, I will focus on the RNAscope multiplex V2 assay. But for now, let's look at some of the applications of the RNAscope technology. As I mentioned, we have been widely applied for detection of long non-coding RNA, splice variants, CDR3 sequences, GPCRs, and so on. And because our flexibility of application, we have been used for basic science research in neuroscience, stem cell, and developmental biology. We have also been used for infectious disease research to identify nucleic acids of emerging pathogens and study infection-induced inflammation. Then we are also used in biopharma discovery and development to assess the preclinical safety and specificity of certain cell and gene therapies. And finally, we have been used in cancer and clinical research. For the purpose of today's presentation, I'll be focusing on the applications of RNAscope in tumor biology and immuno-oncology. RNAscope has been widely used to study different aspects of cancer research. On the screen are listed a few applications of RNAscope in oncology. RNAscope can be used to visualize tumor heterogeneity, to characterize immune cells and soluble factors, to visualize specific CDR3 regions on TCRs, then to detect splice variants of certain targets implicated in cancer, identify non-coding RNAs important in cancer progression, spatially map and validate single-cell RNA sequencing data as it relates to cancer, and detect engineered immune cells within animal models. Let's start with an example of how RNAscope can be used to visualize tumor heterogeneity. In this particular example, we wanted to look at the expression of PD-L1 mRNA in lung cancer tumors. As you can see here, we have compared expression of PD-L1 in 4 different lung cancer tumors. And the expression of PD-L1 varies dramatically based on the different samples. As you can see, the expression of PD-L1 is significantly higher in sample 2B2 compared to 2C4. Now, this highlights the heterogeneous expression of PD-L1 between different tumor types. Interestingly, PD-L1 not only shows inter-tumor heterogeneity, but it also shows intra-tumor heterogeneous expression, by which I mean the expression of PD-L1 varies based on the region within the same tumor, where you can see in the box highlighted on the top of the image, the expression of PD-L1 is significantly higher compared to the box that is highlighted in the bottom part of the image. So this shows that different cancer cells within the same tumors can have different degree of your target gene expression. And these subtle differences in expression can be important when you're studying markers like PD-L1, PD-1, which are important immunotherapy targets. Here's an example of how this group of researchers use RNAscope to study the heterogeneity of Wnt ligand expression in this pancreatic ductal adenocarcinoma organoid library. They wanted to study the expression of Wnt ligands and establish the expression of these ligands, the differential expression of these ligands between normal pancreas compared to a Wnt positive PDAC tumor and a Wnt negative PDAC tumor. And as you can see, the expression of Wnt10A, Wnt7B, and Wnt2B varies between different tissue types, where the expression is highest in the Wnt positive PDAC tumors, whereas the expression is very low to negligible in the normal pancreas. Next, I will talk about the use of RNAscope in characterizing immune cells and soluble factors like cytokines, chemokines, and so on. A lot of the times, researchers are interested in visualizing RNA and protein simultaneously on the same section of the tissue. And with the RNAscope technology, we allow for a workflow where you can perform your ISH assay, followed by which you can perform IHC or IF for your protein target. And this becomes especially significant in immuno-oncology, where you would want to detect specific immune cell types using your antibody of interest, and then visualize gene expression within these immune cells using RNAscope ISH. And here's a great example for the same, where in the cervical cancer tumor, we used the CD3 antibody to mark all the T cells seen in green, and then used RNAscope to visualize expression of PD-L1, PD-1, and interferon gamma within these cells. Now, if you can see the cell marked in the yellow arrow, that's a T cell expressing high levels of interferon gamma. And using this joint workflow, we are able to identify the active immune cells within a tumor microenvironment. Next is an example of how researchers have used this dual-ISH IHC workflow to advance their oncology research. In this paper published in Nature last year, this group of researchers wanted to study the soluble factor LIF and its effect on pancreatic cancer growth. As you can see, using the duplex assay, they were able to show that the LIF is expressed by the stellate cells in the stroma, and they exert their effect in a paracrine action on the tumor cells marked by keratin-19. They show this again by using our multiplex assay and visualize multiple markers simultaneously. Here they have used the dual-ISH IF workflow, where they've used the keratin-19 antibody to mark the tumor cells, and then visualize the LIF factor in combination with CD45 in the stroma. They also show that this LIF is significant in promoting cancer growth by showing that the level of this LIF was higher in cancer patients, in the blood of cancer patients compared to normal patients, normal individuals. They also show a strong association of LIF mRNA with decreased disease-free survival in the patients. So using RNAscope, they were able to visualize the specific stromal expression of this LIF factor and understand how this influences growth of pancreatic cancer. Next, I'm going to show you an example of how, using our BaseScope assay, you can identify specific CDR3 regions on TCRs. Using our BaseScope assay, you can design specific probe against the hypervariable CDR3 regions on TCRs, which can help you identify specific subpopulations of T cells. Now, this is a great example from a paper that was published by Dr. Balco's lab at Vanderbilt, where they were treating a patient with anti-PD-1 therapy. And this patient started showing adverse events due to this therapy, causing encephalitis in this patient, which was ultimately fatal. Now, this group wanted to study what induced this encephalitis and identify the cell population responsible for inducing this encephalitis. So when they performed TCR sequencing, they identified a sequence that was predominantly expressed in this encephalitis tissue. They designed a specific BaseScope probe against this sequence and identified or localized this TCR within the encephalitis tissue. Interestingly, using BaseScope, they were able to co-localize this TCR with the CD4 population of T cells and not CD8 population of T cells, indicating that the immune cell population inducing encephalitis was a CD4 population T cell. Later, they were able to identify that indeed it was a CD4 positive memory T cell population that was responsible for inducing this encephalitis in the patient. So using our BaseScope assay and specific probes against TCR, they were able to visualize this population and co-localize with the right immune cell marker to identify the exact population of T cells. In this other example, this group of researchers was studying the gamma delta T cell population and the changes in this T cell population as it relates to potentiating tumor growth. They were able to show that with age, there was a change in the gamma delta T cell population, and there was an increase in IL-7 expressing cells with age. So if you compare here, mice that were older showed higher expression of IL-7 expressing gamma delta T cells compared to younger mice. And this was directly associated with increased tumor growth in this animal model. Next, I want to show you an example of how RNAscope has been used to detect engineered immune T cells by using an example of our collaboration with Bluebird Bio. Bluebird Bio was developing a CAR T cell therapy against the BCMA antigen and the ROR1 antigen. And they wanted to use RNAscope to study the efficacy and the safety of these CAR T cells. By designing a specific probe against the 3' UTR of the CAR vector, they were able to visualize these CAR T cells within the animal model. Similarly, they designed RNAscope probes for Granzyme B to visualize the engagement of these CAR T cells. As you can see in this particular image, this particular animal was given the anti-BCMA CAR T therapy, and in this particular xenograft of the animal model, you can visualize the BCMA antigen, and you can also visualize the CAR T cells here in red. Now, because the antigen was present, you can see that these CAR T cells have engaged with these antigen-presenting tumor cells, and you can see high expression of Granzyme B, telling you that these CAR T cells are highly specific for BCMA and show an engagement in presence of the antigen. On the other hand, the anti-ROR1 CAR T therapy was not as safe. As you can see here, in this mouse lung tissue, there is some basal level of expression of ROR1, and as a result of this, these CAR T cells were engaging with these cells in the lung and causing toxic effects. Now, when they tried to use IHC to understand why they were seeing lung toxicity, they were not able to see any ROR1 expression. But by using the RNAscope assay, they could see that there was some basal expression of ROR1, which was causing these CAR T cells to engage and give a on-target but off-tumor effect. As a result, this therapy is not safe and was causing significant toxicity. Next, I want to show you an example of how RNAscope has been applied to spatially map and validate single-cell RNA sequencing data in tumors. Now, single-cell RNA sequencing has emerged as a valuable technique to analyze complex tissues and assess the heterogeneity within these tissues. But what you do not get out of this technology is the spatial and morphological resolution. And as a result, it is a very popular workflow to perform single-cell RNA sequencing and then validate some of the key targets using RNAscope. In this particular example, this group of researchers from MIT and Harvard used RNAscope to validate certain key targets in glioblastoma. Using single-cell RNA sequencing, they were able to identify 4 different cellular states in which glioblastoma cells exist and identify specific markers associated with each of these cellular states. When they visualized these markers using RNAscope, they saw that some cells expressed markers for more than one cellular state, indicating that certain glioblastoma cells can exist in a hybrid state, showing phenotypes of more than one cellular state. And this was important discovery to understand how these glioblastoma cells behave in situ and potentially develop a successful therapy against this deadly cancer. So using RNAscope, they were able to validate and also visualize these hybrid states of tumor cells within this glioblastoma. Next, I want to talk to you about the application of RNAscope in visualizing splice variants that are implicated in certain cancers. Here's an example of how researchers used the BaseScope assay to visualize delta 16 HER2 splice variant in patient samples and stratify patients for Herceptin therapy. Tumors that have the delta 16 HER2 splice variant are susceptible to Herceptin. So using the BaseScope assay, they designed probes that would specifically identify the delta 16 splice variant and differentiate that from the wild-type HER2 receptor. If you can see in the panel, on the left-hand side, you have the HER2 IHC, which is not able to differentiate between the wild-type and the delta 16 variant, whereas the RNAscope assay was able to differentiate between the delta 16 splice variant and the wild-type. As a result, they were able to stratify patients based on the RNAscope, the BaseScope signal from this analysis. One of the final applications that I want to talk to you about is the detection of long non-coding RNAs using RNAscope. Here's an example of how researchers use the RNAscope assay to visualize a large nuclear variant of LeuCat1 long non-coding RNA. As you can see in this panel on the top, the expression of the nuclear variant of LeuCat1 long non-coding RNA was upregulated under hypoxic conditions compared to normoxia. And this phenotype promoted an aggressive growth of lung adenocarcinomas in this animal model. In this second example, these researchers wanted to study the expression of this long non-coding RNA in the breast cancer brain metastasis. As you can see here, they compared the expression of this long non-coding RNA between breast cancer that had brain metastasis compared to breast cancer that was negative for brain metastasis. And the expression of this long non-coding RNA is significantly upregulated in the sample that had the brain metastasis, indicating that this particular long non-coding RNA is a prognostic marker for indicating progression to brain metastasis. Next is an example of a few long non-coding RNAs that we studied internally to show that our technology is able to not only detect long non-coding RNAs, but differentiate between the subcellular expression of these long non-coding RNAs. For example, you can see that the MALAT1 long non-coding RNA showed a nuclear expression, whereas the GAF5 long non-coding RNA present in breast cancer tumors showed both nuclear and cytoplasmic expression. So using this technology, you can detect your long non-coding RNA of interest and also study their subcellular localization, which can provide significant clues as to the function of the long non-coding RNA and their prognostic value. We have been applied across various research and application areas, and the adoption of our technology is clear from this graph, which indicates the number of publications that we have using RNAscope. Currently, we have over 2,700 publications, and they have been published in different research areas, with cancer, neuroscience, and infectious disease being our top 3. Next, I want to talk to you about a very interesting collaboration of ACD Biotechnology with NanoString to combine our RNAscope and GeoMx DSP technologies. Here is an image of the workflow wherein you can use RNAscope as a molecular guide to characterize specific regions in the tissue to perform digital spatial profiling. We wanted to show the application of this joint workflow using a specific example. So we decided to study the immune cell infiltration within lung cancer tumor microenvironment. So for that, the step 1 was to identify markers to perform RNAscope on the lung cancer tissue. Since CD3 and CD19 are the most common immune cell markers that would detect T cells and B cells respectively, we use that as molecular guides to perform RNAscope. As you can see here, the expression of the CD19 and CD3 markers was varied dramatically based on different regions within this non-small cell lung cancer tissue. And we decided to profile 25 different regions of interest with variable CD19 and CD3 expression using the GeoMx DSP. So the next step was to profile the selected ROIs using GeoMx DSP. Once we did that, we were able to identify a set of genes that were differentially expressed between the CD19-rich ROI versus the CD3-rich ROI. And the final step was to select some key targets analyzed from the GeoMx analysis and visualize them using RNAscope on a serial section. So for that, we decided to pick 2 ROIs, ROI 6 that was rich in CD19 and ROI 11 that was rich in CD3. We decided to compare the expression of CD8 interferon gamma, CXCL9 and CCL5 between these 2 ROIs. As you can see here, the expression of all these 4 targets was dramatically high in the ROI 11 compared to ROI 6. Now, ROI 11 is the CD3-rich or the T-cell-rich region, and as a result, we would expect the expression of these targets to be high in this ROI, since most of them are T-cell markers. Interestingly, the expression pattern seen here using RNAscope was concordant with what we observed with the GeoMx DSP analysis, again showing that these 2 assays are concordant and show similar expression analysis. RNAscope also allows you single cell and single molecule detection, as observed here in these individual channel panels. So in summary, using the RNAscope, we were able to identify specific regions of interest. Using GeoMx DSP, we were able to profile these regions of interest. And then finally, after identifying key targets, we could perform RNAscope again to visualize these targets using morphological and single cell resolution using RNAscope. So with these 2 technologies, we really wanted to combine the unique capabilities of the GeoMx DSP in providing high-plex profiling of target genes, and then use RNAscope to provide that single cell resolution with morphological context. So with this, we hope to provide a complete solution for gene expression analysis of heterogeneous tumors using single cell resolution. Next, I just want to take a couple of minutes to show how ACD Biotechnology is supporting the advancement of COVID-19 research. We have COVID-19 specific probes for RNAscope analysis. We also have probes specific for the receptor and protease expression. You can study the immune response by visualizing cytokine and immune cell expression. And finally, we have marker probes to visualize the pathological and physiological markers to study the homeostasis of infected tissues. We also have a lot of material available online for people who are interested in knowing more about our joint workflow. We will also be presenting a joint poster at the upcoming AACR meeting in June. With that, I would just like to highlight that in this current environment, a lot of us cannot go back to labs to continue our research, but at ACD Biotechnique, we have a dedicated group of researchers that are working on performing your experiments for you and provide you the results and data. We call them pharma assay services for our biopharma customers and academic assay services for our academic customers. So for more information, please reach out to me or reach out to our technical support and find out about this. Thank you.
Thank you, Anushka. That was a really wonderful presentation. We have a number of questions for you now. The first one is, how can you be sure that one dot is equivalent to one transcript?
So that is a very interesting question. In our technology paper that was published in 2012, we have shown how one dot correlates to a single transcript of RNA. So please go over that particular manuscript and come back to us with any more questions that you might have.
Anushka, another question for you here. Can the RNAscope geomics workflow be performed only using the Leica automated platform?
The RNAscope geomics joint workflow can be performed both in a manual as well as an automated platform. The automated platform is performed on Leica, but we do have a joint workflow that is available for manual assays as well. So you can perform it both using manual as well as automated assays.
And finally, Anushka, your third question is co-detection of RNA and protein using RNAscope and IHC possible on fresh frozen samples?
The dual-ish IHC workflow or co-detection of RNA with protein can be performed on FFPE as well as fresh frozen tissues. So yes, it can be performed on fresh frozen samples as well. Using the RNAscope assay for oncology research, you can understand the complex spatial relationships among different cell types within the TNE. You can profile immune cells and checkpoint markers that are important therapeutic targets. And you can characterize immune cell types and subtypes and study their activation states by visualizing soluble factors like cytokines and chemokines using our technology.